Welcome to the University of Melbourne Biology series. This is Episode 1, The Foundations of Life and the Chemistry of Water. Before we touch a single molecule, sit with one deceptively simple question. Look at a rock, then look at a frog. Both are made of atoms, both obey the same physics, yet one is alive and one is not. What is the difference? Biology begins exactly here, at the line between the living and the non-living. Over the next 12 minutes we will draw that line, trace where life came from, see how we organize its staggering diversity, and then go beneath all of it to the chemistry, and the single molecule, that makes every bit of it possible. Let us begin. So what does it take to be alive? Biologists do not rely on a single test, instead they look for a cluster of shared features. A living thing is built from a common set of elements and organized into cells. It carries genetic information, and it grows and changes over its lifetime. It responds to its environment, and it uses molecules while building new ones of its own. It extracts energy and puts that energy to work. And crucially, life does not exist as lone individuals, it exists in populations that can evolve across generations. No single item on that list is enough on its own. It is the whole package, appearing together, that marks the boundary between a frog and a stone. Notice that cells kept appearing in that definition. That is no accident, and it brings us to one of biology's foundational ideas, cell theory. It makes 4 claims. First, the cell is the fundamental unit of life. Second, every living organism, from a bacterium to a blue whale, is built from one or more cells. Third, cells do not arise from nothing, every cell comes from a pre-existing cell. And fourth, all modern cells trace back to a single common ancestor. That last claim is profound. It means the life you see today is not a set of separate inventions but one continuous, unbroken lineage of cells dividing into cells, stretching back billions of years. If every cell comes from a previous cell, we are forced to ask the obvious question, where did the very first one come from? This is the problem of the origin of life, and it remains an open area of research. There are 2 broad ideas. The first is that life formed spontaneously here on the early Earth, as simple organic molecules assembled into something self-sustaining. The famous Miller and Urey experiment showed that such organic building blocks really can form under early Earth conditions. The second idea looks outward, that life, or at least its ingredients, arrived from space, delivered by comets or meteorites. We do not yet have a final answer, but both routes begin with the same raw material, organic molecules. However it started, life took hold remarkably early, arising soon after liquid water appeared on the planet, more than 4 billion years ago. The first cells were simple prokaryotes. Far later, around 2.1 billion years ago, the more complex eukaryotic cell emerged. And evolution did not just add organisms, it rewrote the planet. Early photosynthetic cyanobacteria converted carbon dioxide into glucose and released oxygen as a byproduct. As that oxygen built up, around 2.4 billion years ago, ultraviolet light split some of it into reactive radicals that combined into a protective ozone layer. This single shift was transformative. It made aerobic respiration possible and shielded the surface enough for life to eventually colonize the land. For most of this history, life stayed small and relatively simple. Then, about 541 million years ago, something dramatic happened, the Cambrian explosion. In a geologically brief window, life diversified at an astonishing rate, and most of the major animal groups we recognize today first appear in the fossil record. We know this period so well precisely because it left behind unusually well-preserved fossils, giving us a vivid snapshot of bodies and body plans suddenly proliferating. The Cambrian explosion is a useful reminder that evolution is not always a slow, even crawl, it can move in bursts, reshaping the living world over a comparatively short stretch of deep time. With billions of years of diversification behind us, how do we make sense of all this variety? We start at the broadest level, with the 3 domains of life. 2 of them, bacteria and archaea, are prokaryotic, their cells lack a nucleus and membrane-bound organelles. The third domain, eukarya, is eukaryotic, with a nucleus and internal compartments. Here is the surprise hidden in that scheme. You might expect the two prokaryotic domains to be each other's closest relatives, but they are not. The archaea are actually more closely related to us, to eukarya, than they are to bacteria. The deepest split in the tree of life is not where appearances would suggest. Within the eukarya we can zoom in one more level, into 4 kingdoms. The plantae are multicellular autotrophs wrapped in cell walls. The animalia are multicellular heterotrophs without cell walls. The fungi are multicellular heterotrophs too, but they keep a cell wall. And the protista are the unicellular eukaryotes, some feeding as autotrophs and some as heterotrophs. To represent how all these groups connect, biologists draw phylogenetic trees, diagrams that map evolutionary relationships and show how lineages diverged from common ancestors over time. These trees can defy intuition. On the eukaryotic tree, fungi sit closer to plants than to animals, a relationship you would never guess from how they look. Let us step inside the simplest of these cells, the prokaryote. Strip a cell down to its essentials and 4 features remain. A cell membrane wraps and contains it. Its genetic material sits loose in a region called the nucleoid, not bound inside a nucleus. The interior is filled with cytoplasm. And studded throughout are ribosomes, the machines that build proteins. What a prokaryote lacks is just as telling: there are no membrane-bound organelles inside. Around the outside, many prokaryotes add extra hardware, a tough cell wall, a protective capsule, whip-like flagella for movement, and pili for attachment. It is a compact, efficient design that has thrived for billions of years. That compact design throws the contrast with eukaryotic cells into sharp relief. The headline difference is compartmentalization. Eukaryotes pack their interior with membrane-bound organelles, while prokaryotes have none. Their genetic material is handled differently too. In a prokaryote the DNA floats free in the cytoplasm, in a eukaryote it is packaged onto chromosomes inside a nucleus. Even their cell walls differ, with the prokaryotic wall built from peptidoglycan while eukaryotic walls vary from kingdom to kingdom. And down at the level of the protein factories, prokaryotes run smaller 70S ribosomes against the larger 80S ribosomes of eukaryotes. Same fundamental job, 2 very different scales of engineering. We have described life from the outside in. Now let us go beneath biology entirely, into chemistry, because every structure we have met is assembled from just a handful of elements. 6 of them dominate living tissue: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. These are the raw material for every macromolecule. But raw atoms are nothing without the bonds that join them, and bonds come in type set by how strongly each atom pulls on shared electrons, a property called electronegativity. When one atom essentially takes electrons from another, you get an ionic bond. When 2 atoms share electrons but unequally, you get a polar covalent bond. And when they share perfectly evenly, the bond is nonpolar covalent. Bonds hold the atoms within a molecule together, but life also depends on the attractions between separate molecules. These are the intermolecular forces, and 3 of them matter here. Dipole-dipole forces are the attractions that occur between polar molecules. London dispersion forces are the attractions present between nonpolar molecules. And the third, the hydrogen bond, is a special kind of dipole-dipole interaction that specifically involves hydrogen. None of these are bonds within a molecule, they are attractions between one molecule and the next. Yet they play crucial roles in the structure and function of biological molecules, and they set the stage for the single most important molecule of all. That molecule is water. A water molecule can form hydrogen bonds with its neighbors, and from that one ability flows a suite of remarkable properties. Water has an unusually high specific heat and a high boiling point, both traced directly to that hydrogen bonding. Its molecules also cling to one another, a property called cohesion, and cling to other surfaces through adhesion. Cohesion and adhesion are important to the way water behaves in biological systems, and together all of these properties make water an essential component for life on Earth. This is why life arose soon after liquid water appeared and never left it behind. From the definition of life, through cells, domains, and chemistry, down to a single hydrogen bond, the foundations all connect. That is the ground we will build on next time.